So, phosphohexose isomerase, does any of you remember what the intermediate is for this? This has a very specific intermediate that we'll see again.
Is that the one that opened up?
Well, yeah, it starts off as a ring, but that's not the intermediate we're looking for, right? So let's go to here. So here we have the ring opening. Does this thing have words? Probably not. So this is ring opening. So is that what you were talking about, opening up? Yeah. Do we need to continue? Okay, so there we have the aldose now of glucose with the free aldehyde, and that can be then reactive. So what do you think happens next? Yeah, but we get rearrangement. This is a rearrangement. So because it's a isomerase, so we're taking 6-phosphoglucose and making 6-phosphofructose. So we need to, instead of an aldehyde, we need to turn it to a ketone. So do you remember what happens next? There's going to be a base, right? Base picks off. Because if you notice, they have this hydrogen all out here with its little electrons there. So a base is going to kick that off, right? So base picks that off. Picks off that hydrogen. These electrons have to go somewhere they go down, and then you form an oxide, which can then pick up a hydrogen. Yeah. But then do you know what this is called, this intermediate? That ene diol, remember this is an ene group where you have an unsaturated bond between two carbons, or a double bond between two carbons. And diol, if you had a hydrogen here, it would be an alcohol, so two alcohols. So ene diol intermediate, right? And then that continues. So you see it becomes the ene diol there. And then these electrons drop back down, and the electrons that were forming the ene pick off the original hydrogen off that enzyme, off the base, off the enzyme. And now you have 6-phosphofructose, right? So let's see what's next. I think it's going to show you a ring closure here at the end. And do you guys have access to these online?
Where is it located? Or where would it be located?
So let me see. Let's just check this out. Just allow this active content. So let's see if we can't find these online. I think you might have access to all these animations, but maybe not. Here we are. So this BCS Freeman site, so they have these free and open resources, and so the student resources are here. Let's see what we have here.
Be under the molecular structure tutorials. Up the top. Down at the Raspberry resources. The fourth resource.
Oh, okay. Lecture and companion art, living graphs, web links, and molecular structure tutorials. Yeah, I don't know if you guys have the animations per se, but here they have everything else. So if we go to the link, they have web links and then the companion art, and that companion art's going to be like a big, probably like 30 megabyte file that you'll download with all the pictures, and then the web links. So they have a lot of web links. Let's see, the Triose phosphate isomer.
Oh, is this for the project thing? Or?
Huh? No, this is the books, student resources.
Oh, okay.
I was seeing if they had the animations. We just did one animation. We'll go back to, because I forgot to put these on this computer. We just did now we'll do the aldolase. Okay, so we haven't reached this one yet. But this is the next reaction we're going to cover in the PowerPoint. So we might as well just go through this one here, right? So can anybody tell me what happens during the aldolase reaction? So we start off with fructose 1,6-bisphosphate because the last reaction was which enzyme? You'd mentioned it a couple times before. Kinase, yeah, 1. So that's very important to distinguish between phosphofructokinase 1 and phosphofructokinase 2. Because in the next chapter, we'll be learning about phosphofructokinase 2 and its functionality. It's like a bifunctional enzyme that also has 1,6-bis-fructose phosphatase activity. And so it's involved with gluconeogenesis, and it makes what, 2,6-bisphosphate, glucose 2,6-bisphosphate rather.
Are you saying that phosphokinase was in the last mechanism?
No, so the last mechanism we saw was the isomerase.
Yeah.
The next step in glycolysis is the phosphofructokinase, and that's just adding a phosphate. So you get attack of that number 1 hydroxy on the fructose attacks that gamma phosphate on ATP, and that's basically the mechanism. So, but for this, it's a little more complicated. This is probably the most complicated mechanism you'll see here in glycolysis, and I think the hardest mechanism is going to be your pyruvate dehydrogenase mechanism, which is the same mechanism but just a different substrate, alpha-ketoglutarate dehydrogenase. It's part of the TCA cycle. So, yes. Okay, so here we have fructose 1,6-bisphosphate that was just produced by phosphofructokinase 1. And again, we get ring opening, so because we're going to have the free ketone then that's going to work with. So here we have fructose as the ketone form. And it's going to go into the enzyme like this, and here we have an active site lysine. So this is a real interesting mechanism. Then we can hit continue. So the free electrons on the nitrogen on the lysine are going to attack the carbonyl on the ketone, like that. So that's an intermediate. If we continue, you have a quaternary nitrogen then here. This can then form this quaternary shift base type structure, right? So you have a protonated shift base, okay? And can anybody tell me what's useful about quaternary nitrogens? What are they? They have these great electron withdrawing properties, right? Because these electrons can go here, and then it'd be like a normal nitrogen. But then they're going to have to come from somewhere, right? This carbon will then be lacking electrons. So we'll continue. So we get a base, picks off this hydrogen, this falls back down, and you're going to form an aldehyde. These electrons have to go somewhere. They're going to form this ene group between these two carbons, and then the quaternary nitrogen then, can suck up that extra electron, and then all is good. And if you notice, we've cleaved that carbon-carbon bond between 3 and 4. We've made glyceraldehyde 3-phosphate, and then we still have this 3-carbon adduct bound to the enzyme. And then the rest of the mechanism here is pretty simple. You have covalent enzyme enamine intermediate.
When you get the cleavage between the two, is it due to the fact that you've got equal polarities on both sides and just slices down the middle?
Equal polarities on each side. So what's happening is the base picks off the 1 hydrogen on the alcohol. There's electrons there. They have to go somewhere. Or they don't have to because oxide is okay too. But anyways, those electrons drop back down because you have this electron withdrawing properties on the shift base.
Right.
They're the quaternary, protonated shift base. So the push from the oxygen, the electrons kind of want to go down, and at the same time, this quaternary nitrogen is great at withdrawing electrons, so it kind of pulls the electrons at the same time they're being pushed.
Okay.
And then you get
That's
split.
why it separates into three.
Yeah. So another protonated shift base, and here water just comes in and replaces that nitrogen. Then those will drop back down. Another base will pick off that hydrogen. And then you get this is dihydroxyacetone phosphate. So the other 3 carbon sugar that's split from the, splitting fructose into 2 sugars. Okay?
I have a question. When we draw our mechanisms, how do you want us to draw the enzymes like that? Just the groups like that? Or?
Yeah, that's fine.
Yeah.
Unless I told you about a specific cofactor.
Okay.
So, no, I want you to draw all the amino acids with all the side chains and where they're located in 3-dimensional structure.
I'll take the pictures of that.
No, like that's really normal how you're going to see these. So then that's that for the aldolase reaction. So aldolase, you split it into glyceraldehyde 3-phosphate and then dihydroxyacetone phosphate. Now you got these 3 2-carbon sugars. Do you know what reaction is going to be next? Anybody? It's got a familiar,
Separate from the azoles?
huh?
You separate from the azoles?
So we have the 2 different 3-carbon sugars. One's an aldose and one's a ketose.
Oh, we have to convert.
Yeah, so you have to convert one to the other. So what kind of intermediate are you going to see? See, you weren't here when we went over the other isomerase. Do you remember?
Phosphohexose?
Well.
You're talking about I jumped in.
So the next reaction is not this reaction. Well, this is what we just looked at, but it's not this reaction. But it's another isomerase reaction, right? So we go from the dihydroxyacetone phosphate to the glyceraldehyde 3-phosphate again. So we're switching from a ketone to an aldehyde again.
Right, we get 2 G3P. I'm writing that down.
Yeah. So we switch from the where are we? I think here. So this is the aldose cleavage. Here we go. No, that's aldose again. So triose phosphate isomerase. So this is basically the same reaction with different products and substrates as the hexose phosphate isomerase reaction. So do you know the intermediate? Anybody? Anybody? What's that guy's name in that movie?
The hexose phosphate?
Okay, that guy.
So yeah.
The
So this is triose phosphate isomerase rather than hexose phosphate isomerase. Same intermediate, same basic mechanism.
Bueller.
Yeah, Bueller. That's what I was thinking of. I can't remember. So again, the rationale behind this is glycolysis to proceed by one pathway. Aldolase makes these 2. Only GAP is the substrate for the next enzyme. So GAP-DH is the next enzyme. So dihydroxyacetone must be converted to glyceraldehyde 3-phosphate. And this completes the prep phase of glycolysis, and then it's thermodynamically unfavorable, so it's reversible. So what do we know about the unfavorable reactions?
They don't occur. They don't occur.
No, they do, but what drives them? Basically, delta G is right around 0. So what's going to drive it to go one direction or the other is a concentration. So it's going to be based on which, what's going on in the cell. If you're burning energy, you'll do glycolysis. The same enzymes used in gluconeogenesis. So if you're making sugar, it'll run in reverse. Okay? So it's just, depends on the concentration of the substrates and products at the time, which direction it's going to go. But in glycolysis, the gap concentration is kept low to pull that reaction forward. So glucose, carbons, and gap. Oh, this is that carbon counting thing. So don't worry about counting your carbons like that. That's like core bio stuff, right? Oh, they totally skip over this mechanism. But basically it's the same mechanism, right? As before. Can somebody explain it to me? So okay, so what do we have here? Can anybody tell me what's going to happen? Yeah, so this base will take this, and then these are going to what? Wait, did I do this wrong?
The base takes a hydrogen, then the oxygens are left. The electrons left to the oxygen.
Yeah, I think I did this wrong, right? What did I do? Now I can't remember the darn mechanism. We can see it. Yeah, this mechanism basically, but instead you only have these three. Oh, I had it pick off the wrong darn hydrogen, right? So. So now the base takes this, right? See how easy it is to make mistakes? So these have to go down, and then these will go up. And they'll eventually pick up a hydrogen. And then what's the intermediate called?
Cis and no.
Don't worry about the cis, but yeah. Just ene diol. Because remember the double bond is an ene group, right? From organic chemistry. Double bond's an ene, so you have this ene diol intermediate. So diol meaning there's two alcohols. Okay. So same mechanism for the triose or phosphotriose isomerase. Okay? With that ene diol intermediate. Now we'll do glyceraldehyde.
Wait, so what was that?
That was the triose phosphate isomerase. So a base picks off a hydrogen, electrons drop down to form the ene group, and then these electrons have to come up and they'll pick up a hydrogen ion. So you have an ene diol intermediate. Okay? Why are you looking at me like I'm crazy?
No, I'm not feeling good. So
That's
like.
that's okay. Sorry.
Clearly your fault.
So next we have oxidation of glyceraldehyde 3-phosphate. And I think we have this again then. So here we have the mechanism here. Glyceraldehyde 3-phosphate dehydrogenase, or GAP-DH. When you take methods 2, this is what you're cloning. Okay, so in this, so we have an active site, cysteine, that's going to form a covalent bond with your intermediate. And then what's NAD?
Nicotinamide. Adenine.
Dinucleotide. Even though it's a single nucleotide, they got those crazy things. And So if you guys remember the nicotinamide, I'm not good at drawing. What am I doing? Like this. So that's charged, right? And then I think there's an R group here, and then this goes somewhere else. So where is this, so what is this gonna do, this nicotinamide? Adenine.
Take out the hydrogen.
Hydrogen, not a hydrogen ion, but it is a hydrogen ion, but a hydride, right? So it takes off a hydride, and where does it go here? Is it gonna go here? These aren't the right numbers, but which number that I just made up would it go to? Yes, so it's gonna go to 3. You'll get, these electrons will go here, these will go here, and then these will there. So you lose aromaticity, but you also lose the charge. So, and you picked up a hydrogen. It's been reduced at that point. So, okay. So yeah, they don't show you the structure of NAD there, but I might ask about it on the test, so I put it covering it once again. Okay. So, where are we? So we'll play this. There's your glyceraldehyde 3-phosphate. Comes in, binds at the active site. If we hit continue, the sulfur, the sulfhydryl on the cysteine attacks that carbonyl. You get a quaternary structure. And then what's going to happen, those electrons drop down, and that allows this hydride to be picked up by the NAD right here, right at the top of that NAD structure. And then what's going to happen? Oh, I don't hit that button, I hit this button. So then the NAD diffuses away, and a phosphate's going to come in. A phosphate attacks that carbonyl and replaces that sulfur adduct, and you have 2,3-bisphosphoglycerate, or not 2,3, 1,3-bisphosphoglycerate, right? So you've oxidized that glyceraldehyde to a carboxylic acid, and now you have glycerate. So that 8 means it's a carboxylic acid, and it's a phosphoanhydride of that glycerate, because you used a cysteine, bond there is a high energy bond, but you've replaced it with a phosphate, so this is another high energy phosphate. So, okay. So the next reaction, do you know what the next reaction does, anybody?
NAD's there, so it's got the hydrogen.
Well, now this one, we're done with that one. And we're not even on the next one there, so we got to go back to the other PowerPoint. So this is what we've done now. We have 1,3-bisphosphoglycerate. And here's the mechanism all spelled out again in individual forms. So I think you have these in your book, right? Mechanism of GAP-DH substrate binding and so forth. So the whole thing, they go over and over again with the same slide over and over again. Okay? So now we have the first production of ATP. So you have this high energy phosphate, 1,3-bisphosphoglycerate. It's not the highest energy phosphate that we see, but it's the second highest in glycolysis. So this is much higher energy compound than ATP. So it's able to donate that one of its phosphates to ADP to make ATP. Okay? So basically, it's a simple mechanism also. And I don't think they show you the mechanism, basically, but it's phosphoglycerate kinase, heavily favors the formation of ATP. And this is one of these enzyme names that kind of runs backwards, because normally when you think of kinases, it tells you what gets phosphorylated. So we're not phosphorylating phosphoglycerate, we're actually phosphorylating ADP to make ATP. So, but it's heavily favored to run in this reverse direction from what it's named, basically. Okay. And then we have the substrate level, and this is, did I mention, this is called substrate level phosphorylation. And that's when you're taking the phosphate group directly from one compound and adding it directly to a ATP. Okay. And kinases are enzymes that transfer phosphate groups from ATP to various substances, and this one kind of runs in reverse because it's actually phosphorylating ADP to make ATP. It's highly thermodynamically favorable, and it's reversible because of coupling to get DH reactions. Oh. It's these chairs, they tried to kill me. So the next reaction is this phosphoglycerate mutase. And it's a mutase, so instead of, we're going from 3-phosphoglycerate to 2-phosphoglycerate, and it has a very low change in free energy standard state. So again, that's going to be driven mainly by concentration. And the rationale is to be able to form some high energy phosphate compounds. Mutases catalyze the apparent migration of a functional group, so we're taking it from the 1 and adding it to the 2. And one of the active sites, histidines, is post-translationally modified to a phosphohistidine. So this enzyme already has a phosphohistidine there. And you'll see it's just exchanging that high energy phosphohistidine and adds it to one of the alcohol groups and then reverses that. We'll see. And phosphohistidine donates its phosphate to the O2 before retrieving another phosphate from O3. So there's 2 different sites on these histidines. And let me show you again. So this one we have the mechanism animation for. So you have the active site histidine that has been phosphorylated, and you have another histidine that's in the active site that's not phosphorylated. And what you do, as it slowly plays, 3-phosphoglycerate there. So this nitrogen on the histidine draws that proton away from the alcohol, and then that alcohol is going to attack the phosphate that's already adducted to the histidine at the other site. And then the next step, basically that nitrogen then just attacks this phosphate, and so it's rephosphorylated, and then you've just basically moved one phosphate from here to the other, but this isn't the original phosphate. The original phosphate sticks and stays with the enzyme, and that enzyme's ready to do that to another substrate. And that one we won't see for a while. Okay. And here's the overall mechanism again. So it's a pretty simple mechanism. Next you have dehydration of 2-phosphoglycerate to phosphoenolpyruvate. Basically, you're just going to remove this water and form this ene group, and you have phosphoenolpyruvate at this point, and that's a really high energy compound. And if you notice, free energy fairly low for this. And the rationale is to generate a high energy phosphate compound, which will then be used for another round of substrate level phosphorylation to make another nucleotide triphosphate. So 2-phosphoglycerate's not good enough phosphate donor to do that, to make, ATP from ADP. But 2 negative charges on 2-phosphoglycerate are fairly close, but loss of a phosphate from 2-phosphoglycerate would give a secondary alcohol with no further stabilization. So it's even slightly thermodynamically unfavorable. The product concentration is kept low to pull it forward or in reverse for gluconeogenesis. So then, wait, did we not have the mechanism here? So this is a fairly simple, huh?
At the enzyme, the electrons is pushed to the ene.
Yeah, so you're going to get a base that's going to pick off this hydrogen probably. These will drop down, and then this can come up and pick up another hydrogen. So it's pretty simple. And then you have the ene group and dehydrated that. Or there's another possible mechanism for that catalysis also. So now we have, where are we? Phosphoenol pyruvate. And so this is pyruvate kinase is this last mechanism, right? And pyruvate kinase, again, is a substrate level phosphorylation, so we end up with ATP and pyruvate. So you get energy 2 times out of this reaction, right? So first you remove the phosphate here, it gets picked up by ADP to make ATP, and you're going to end up with the enol form of pyruvate, and that's heavily not favored, so it's much favored if you get rearrangement back to the keto form, right? The keto-enol shift. So the rationale for this is substrate level phosphorylation, net production of 2 ATP and glucose. You have loss of phosphate from phosphoenol pyruvate yields this enol that then tautomerizes into that ketone, and that's heavily favored. This tautomerization effectively lowers the concentration of the reactant product and drives that reaction toward ATP formation. So you're taking away the enol form like almost instantly after it donates that phosphate to make ATP. That's going to really drive that force. So pyruvate kinase requires divalent metals, magnesium or manganese for activity, and it's highly thermodynamically favorable, irreversible, and it's heavily regulated. So it's regulated by ATP, divalent metals, and other metabolites. So there's 3 main steps in glycolysis that are heavily regulated. Anybody know what those are? This is one of them, right? The pyruvate kinase step, hexokinase. Were you raising your hand? Oh, okay. Huh? And then one is the most regulated. Do you know what that is? It's the committed step for glycolysis.
Phosphorylation of glucose?
That's hexokinase. So that's not the committed step to glycolysis. Yeah, phosphofructokinase 1 is the most regulated. And that's the committed step for glycolysis. So you'll see a lot of regulation that goes into that. So, which I was saying, don't get PFK1 confused with PFK2. Because PFK2 actually produces an allosteric regulator for PFK1. So, okay. So this is the summary. Like I told you, I'm not going to quiz you on memorizing the number of things that are like that. So we get 4 ATPs and blah, blah, blah. I'm sure you learned that 2 times in micro and core biology before. But glycolysis is heavily regulated, so it ensures proper use of nutrients and ensures the production of ATP only when it's needed. So, and glycolysis will actually occur at elevated rates in tumor cells, right? But what's the problem if you use glycolysis at elevated rates?
Uncontrolled.
It's not that it's, well, yeah, it's uncontrolled, but you're going to consume something.
But you can put, yeah.
Well, yeah, you can, of course you consume sugar. But what's one of the other things that are necessary to continue glycolysis that you're going to use up? Well, but you're going to make a lot of ATP. You make more ATP than you consume. What don't you use that you produce. Or that there's one thing that you use up that you can't recycle. I mean, you can recycle, and normally it gets recycled in the electron transport chain oxidatively, but in cases of cancer cells or muscle cells working where they don't get enough oxygen, they can't. Huh?
It's not the enzyme.
It's a cofactor for the enzyme.
COA?
Huh? No, the NAD.
I was gonna say.
So the NAD, we need that to do more glycolysis, right? So if cancer cells, for example, they're going to be doing glycolysis like out of control, they'll produce a lot of NADH, and then they'll run out of NADH. They have to could get back some of that NAD. So this is where these fates of pyruvate come in. So for anaerobic conditions, to regenerate that free NAD, you can either produce ethanol by fermentation or lactate. You take pyruvate and you reoxidize it to lactate. Is it oxidized? No, you reduce it to lactate. Reduce it to lactate, and then then in the process of reducing it to lactate, you've reoxidized the NAD, so you have NAD plus again that can then go back and produce more ATP via anaerobic glycolysis. But what do we do with all this lactate? Like, nah. You know when you work out, you get the muscle soreness, they all say that's lactic acid. So, but if you're like in a fight or flight situation, you need to convert that lactate back to sugar. Do you know where that occurs?
Liver.
Yeah. So your lactate gets spit out into your blood, it'll transport to your liver will convert this back into pyruvate and make new sugar out of it and put that back into your blood system. Your liver does the same thing for your cancer cells. So, and this is called the Cori cycle. So, but we'll talk about that after we talk about anaerobic glycolysis from fermentation, and this is what I was just talking about, right? So, without consuming oxygen, generation of energy, ATP without consuming oxygen or NAD+, you have no net change in the oxidation state of the sugars, reduction of pyruvate to another product, regenerates NAD for further glycolysis. Because that NAD is going to be the limiting step for how much sugar you can burn until you need to get it back so you can burn more sugar. So you take the NADH and make lactate or alcohol, and then you'll reproduce or regenerate NAD. So animals undergo lactic acid fermentation, so reduction of pyruvate to lactate, and that's reversible. During strenuous exercise, like we were saying, lactate builds up in the muscles, and it's generally less than 1 minute will start generating lactic acid. The acidification of the muscle prevents its continuous strenuous work. So lactate can be transported to the liver or converted to glucose, and this requires a recovery time, high amounts of oxygen consumption to fuel gluconeogenesis, and restores muscle glycogen stores. So not only will muscle do this, but cancer cells will do this. Because the cancer cells, they'll grow so fast, they outgrow the vasculature growing into that tumor. So they'll do a lot of lactic acid fermentation, send it to the liver, and then that liver will convert it back to sugar, and those cells will take that sugar up again to burn. Anybody know what that cycle is called? It's like a...
Krebs cycle.
No, it's not the Krebs cycle. The Cori cycle. So, and this is the overall reaction. So we have pyruvate. NAD is going to add a hydrogen to this, and you regenerate NAD. So NADH, adds the hydrogen. You regenerate NAD, and then you have lactate, L-lactate there. So, and that's heavily favored. And so for each glucose, you're going to generate 2 NADHs, 2 pyruvates, and then that will equal 2 lactates to regenerate the 2 NADHs. Don't worry, I'm not going to, again, ask you the numbers of things. I don't like that. But this is the Cori cycle, right? So using your muscles, they don't get enough oxygen, you're going to produce lactate. That lactate gets transported to the liver. Gets transported to the liver because the liver has a lot of excess NAD. They can then take that lactate and with ATP make glucose. So it's a very inefficient cycle too. You think you burn more ATP making sugar than you do getting out of it. So you use more energy by producing this lactate, and then at the same time you can ship glucose back to the muscle to make more ATP in the muscle. I think you lose about 2 ATPs for each sugar molecule that you make, and then that just gets reburned again. So it's actually not very efficient, but it gets sugar to where it needs to be burned. So. Then yeast will undergo ethanol fermentation. It's a 2-step reduction of pyruvate to ethanol. It's irreversible. Humans do not have pyruvate decarboxylase, so we do express alcohol dehydrogenase for ethanol metabolism. CO2 is produced in the first step, is responsible for carbonation of beer, dough rising when we're making bread, and both steps require cofactors. Pyruvate decarboxylase uses magnesium, and my favorite cofactor is thiamine pyrophosphate. And alcohol dehydrogenase uses zinc and NAD. So for your mechanisms, I guess it's the pyruvate decarboxylase mechanism will help you out for the pyruvate. No, I can't think. The first step, where you take pyruvate and make acetic acid. It's pyruvate decarboxylase. But the first step in the TCA cycle, before you get to the TCA cycle. So here we go, we have pyruvate decarboxylase using TPP, decarboxylates it, makes acetaldehyde, then alcohol dehydrogenase will convert that, it'll reduce it and convert that to ethanol. So this is the toxic intermediate when you're burning alcohol also. The alcohol dehydrogenase runs this way. It's acetaldehyde. Eventually it'll get converted to acetic acid, but if this sticks around very long, it's toxic. Likewise, if you drink methanol, you take methanol and alcohol dehydrogenase converts it to formaldehyde. That's even more toxic than acetaldehyde and will stick around longer, cross-link proteins and whatnot. So if you ever drink methanol, the way to treat that is to drink ethanol. You outcompete alcohol dehydrogenase with the ethanol and make, that's how you treat methanol poisoning. So. But anyways, this is the mechanism that should help you. It's not the complete mechanism. But here we have TPP. So thiamine pyrophosphate. So if you look at that structure, again, you have this quaternary nitrogen there on that ring structure, the thiazolium ring. So it ionizes. What makes it possible to ionize is that positive charge there makes this hydrogen basic. So that hydrogen can give up that carbon can give up its hydrogen, and it'll stabilize this carbanion. You have this positive charge here and a stabilized carbanion. That carbanion can then attack the carbonyl pyruvate, and you have this adduct with the thiamine pyrophosphate covalently bound, which again, you have a quaternary nitrogen, which is great at pulling on electrons. So this will pull on these electrons, and then you have this carboxylic acid, you have this oxygen that's charged. The electrons will drop down, leaving these electrons nowhere to go, but they can come here because these electrons can, take care of that charge and get rid of that charge on that quaternary nitrogen. And in the process, if you notice, when these electrons fall down, that basically is carbon dioxide that you form there. So now you have acetaldehyde in an adduct form with the thiamine pyrophosphate. This is resonance stabilized. As you can see, these electrons can drop down, and then these can, you have a carbanion there, or you have the ene with the no charge there, right?
Is this a cyclic process that just starts over again?
Yeah, it'll go over and over. So then for this pyruvate decarboxylase, you end up here, this is going to basically acetaldehyde, because what will happen is this alcohol group, the electrons will drop down and it'll kick off, because remember this carbanion is stabilized in this diazoleum ring, and then you have acetaldehyde right there. Now that's not what happens in that mechanism I want you to draw out. What happens in the mechanism that I want you to draw is lipoproic acid actually comes in and attacks here and reduces it or oxidizes. Yeah, because you're adding electrons there. I always say things wrong. But anyways, this is pyruvate decarboxylase. So that should help you with that, the hardest mechanism that we have. So TPP is a common acetaldehyde carrier. So this is the enzyme I was trying to remember. Pyruvate dehydrogenase. Similar to pyruvate decarboxylase. This is what we see in ethanol fermentation. Pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase we see in synthesis of acetyl-CoA and in the citric acid cycle for both. So, and then trans-ketolase will also see it, but okay, so that thiamine pyrophosphate. A fun coenzymes. Next is the gluconeogenesis and the precursor for carbohydrates. So if you notice, mammals, we cannot convert fatty acids to sugars. So if there's a question on the test and it asks like which one of these can be used to make sugar, if you see something that's a fatty acid, that's, you know, which one of these cannot be used to make, you know, glucose. If you see a fatty acid there, that would be the right answer, right? So, and what's an example of a fatty acid? Yeah, phospholipids contain fatty acids there, but they're glycerol phospholipids. Triglyceride has fatty acids and ester groups with the glycerol, but like palmitidic acid, propionic acid, those are both fatty acids. One's long chain, one's short. What's shorter than propionic acid? Acetic acid.
Do we need to know all those on that table?
No, but just know what's a fatty acid and what's not. Now, I'm not going to ask you about all those ones on the table, but if you see palmitate, you know it's the conjugate base form or the charged form of palmitidic acid. Acetate is the conjugate base form of acetic acid, right? The 2-carbon acid.
That's citrate together?
Yep, citrate.
There's a trend there.
So glycolysis versus gluconeogenesis. So glycolysis mainly is going to occur in the muscle and the brain, and gluconeogenesis mainly occurs in the liver. There's actually 2 organs that can do gluconeogenesis the whole pathway, and that would be the liver and the kidneys. And the reason is those are the only two organs that produce that last enzyme in the step, which is this glucose 6 phosphatase. So you remember what phosphatases do?
They lyse. They lyse the glucose.
They don't lyse the glucose, they remove phosphates. There's protein phosphatase 1, removes phosphates from proteins. Glucose 6 phosphatase removes that phosphate from glucose 6 phosphate, making glucose, and that glucose can then be spit out into the bloodstream as free glucose for transport around the body. So that's why you see this in the liver, and the kidney has some glucose 6 phosphatase activity also, but it's mainly in the liver. Okay, but if you notice here, all the enzymes are pretty much the same except for 3, right? And those are the heavily regulated enzymes. The hexokinase has a different enzyme. The phosphofructokinase 1 has an alternative enzyme. Everything else is the same until we get down to the pyruvate kinase is replaced by 2 separate enzymes. So all the steps are the same except for the phosphofructokinase 1, hexokinase, and pyruvate kinase are replaced. So these are both heavily regulated pathways, right? So you don't want to be burning sugar at the same time you're making it, because that would be known as a futile cycle, where you're not really doing anything but just burning energy in the process, because it takes more energy to make the sugar than you get out of burning it, right? Because we're not 100% efficient. That energy gets released as heat that doesn't get, converted back into the chemical energy. So, opposing pathways that are both thermodynamically favorable. They operate in opposite directions. The end product of one is the starting compound in the other. Reversible reactions are used by both pathways. And what drives which direction those are going to go in those reversible pathways?
Concentration.
Yeah, concentration, because the free energy change is really low for those reactions. So those are being driven by concentration, because those reactions are near equilibrium. So irreversible reactions of glycolysis must be bypassed in gluconeogenesis, and these are highly thermodynamically favored and regulated. So a lot of the regulated steps are the ones that are really have a high free energy change. Different enzymes used in different pathways, differential regulated to prevent a futile cycle, like I was saying. So pyruvate to phosphoenolpyruvate requires 2 energy-consuming steps, which means you're going to be using up 2 ATPs to make that phosphoenolpyruvate from the pyruvate. So the first step, you take pyruvate carboxylase. So what do you think pyruvate carboxylase is going to do?
You remove the carboxyl group?
Not quite. It's going to add the carboxyl group. So pyruvate decarboxylase was that removing the carboxyl group to make acetaldehyde in fermentation, right? So we add the carboxyl group to pyruvate, and that converts pyruvate to oxaloacetate. Does anybody remember that from like biology or micro? That's like one of the first entry steps into the citric acid cycle, right? So this is an intermediate in the TCA cycle. Carboxylation uses a biotin cofactor. And you guys haven't learned what biotin is, right? It's a cofactor. It's on a string, basically.
So the difference between the carboxylase and the decarboxylase is it's not the difference between the phosphatase? Is there a dephosphatase right now?
No, so the phosphatase, see, the terminology is a little funky. So a kinase adds phosphates, a phosphatase removes phosphates. And this one might actually be better because a carboxylase adds a carboxyl group. Decarboxylase removes the carboxyl group, right? Yes?
Phosphokinase adds the phosphate group, and then phosphorylase.
Phosphorylase, okay, so phosphorylase is like instead of adding water across to break a bond, or to break a bond, basically, like normally you add water across a phosphate ester bond, and that removes the phosphate. A phosphorylase uses phosphate across that phosphate ester bond to break that. And that's when you're breaking down glycogen. Those glycogens are held together by those alpha 1 to 4 glycosidic bonds. And you use phosphate instead of water to break that. So you end up with, is it glucose 1-phosphate? I think we had this discussion before. Yeah, glucose 1-phosphate you get as a result. And that's already got the phosphate, so it's ready to be burned as sugar. It just gets converted to glucose 6-phosphate before it enters glycolysis. So you're not burning another ATP to phosphorylate that glucose. Second step, phosphoenolpyruvate carboxykinase. So again, phosphoenolpyruvate carboxykinase, so you're going to have phosphorylation in this step. Converts oxaloacetate to PEP, and then the phosphorylation from GTP and decarboxylation is what happens. So GTP is just like ATP, it's just guanine instead of adenine is the handle. And then occurs in the mitochondria or the cytosol, depending on the organism. So first step, synthesis of oxaloacetate. You have bicarbonate and pyruvate. They're going to react along with ATP and biotin to make oxaloacetate. And hope we have the mechanism, because this bicarbonate isn't going to add easily. It has to be, you know, energized or activated. And it's activated via phosphorylation with this ATP. Okay, so you activate that bicarb. And it does this at an enzyme that has 2 active sites, and biotin is like on a tether. It's at the end of a lysine chain and then has a long chain itself, and it can swing from one active site on the enzyme to the other. One site, it picks up the carboxy group. The other site, it delivers that carboxy group and adds it to the pyruvate. So this first, so they don't show you this initial step, but basically at this first step, you produce carboxyphosphate from the bicarbonate, so that carboxy group is phosphorylated, and that activates it to be added to the biotin. The biotin picks it up, you remove that phosphate, gets let go, and then it goes to the next site where you have the pyruvate can then grab onto that to form the oxaloacetate, and they show you that reaction. The next reaction here, where you decarboxylate the oxaloacetate. So here you have the oxaloacetate with that carboxyl group that you just added, and that took you a nucleotide triphosphate to add that. These drop down, these will come over here, causing these to come and pick up the gamma phosphate from a GTP, and then you have phosphoenol pyruvate, right? So additional bypasses, catalyzed reverse reaction of opposing step in glycolysis are irreversible themselves. So fructose 1,6-bisphosphate gets converted to fructose 6-phosphate by fructose bisphosphatase 1. Again, it's a phosphatase. It's bisphosphatase because it's fructose bisphosphate, and it's phosphatase. We're removing one phosphate to produce fructose 6-phosphate. Coordinately oppositely regulated with phosphofructokinase. Okay, and then glucose 6-phosphate gets converted to glucose by glucose 6-phosphatase. So, and these are simple hydrolysis reactions where you add water to substitute for that phosphate group. Phosphate's a great leaving big bulky charged group that gets stabilized by resonance after it leaves. So from pyruvate to phosphoenol pyruvate, again, so this, you know, certain things occur in the mitochondria and certain things only occur in the cytosol. So pyruvate will enter the mitochondria, get converted to oxaloacetate. Oxaloacetate can't be transported out of the mitochondria, so it has to be reduced to malate before it can be transported out of the mitochondria. Then malate can then be reoxidized to oxaloacetate to produce PEP. So there's a bit of a shuttle that you have to go through. And then PEP, yeah, okay, so then PEP basically can then get converted to ATP or sugar, basically, through gluconeogenesis in the cytosol. But you have to get this oxaloacetate, pyruvate kinase occurs in the mitochondria, and so the oxaloacetate has to work its way out of the mitochondria. And it does that by being reduced to malate, because it can't.
Those are two separate mechanisms, right?
Yeah.
And so
They're basically the same mechanism, but here we've converted lactate. So this would be happening in the liver. We take lactate up from the bloodstream. Lactate dehydrogenase converts it to pyruvate. Then pyruvate goes into the mitochondria. And you've noticed these are the exact same thing. Mm-hmm. Except here, the oxaloacetate can be converted to PEP directly in the mitochondria. But then gluconeogenesis occurs in the cytosol, so it has to be transported out also. So no matter what, you have to get the PEP out of the mitochondria to make glucose. Whereas do you know what happens? Why does pyruvate go into the in here to begin with? This is where the TCA cycle is going to occur. Okay? And we'll learn about that, not the next chapter, but the next chapter after this next chapter. So gluconeogenesis is expensive. So it costs 4 ATP, 2 GTP, and 2 NADH, but it's physiologically necessary. The brain, nervous system, and red blood cells generate ATP only from glucose, so we have to have glucose around. When glycogen stores are depleted, we have to be able to make glucose. And we can make glucose from amino acids. From glycerophospholipids, we can make glucose. But can you tell me what part of a glycerophospholipid we make glucose from? I told you we couldn't from the fatty acids. But what other structure is there? Or even triglycerides we can make sugar from, but not, remember, not the fatty acids. So what's the only part left after the fatty acids and triglycerides?
Amides?
Not amides, triglycerides. So what's the backbone for triglycerides? Same backbone for glycerophospholipids. I'll draw it. Huh? Glycerol. So glycerol you can make to sugar. Fatty acids cannot be made into sugar. So, but glycerol and amino acids, a lot of them can be converted to sugar. So
is glycerol like also in your muscles?
Well, yeah, glycerol's everywhere. Where you have membranes, you have glycerol, right? If you notice, some of the intermediates in glycolysis are not glycerol themselves, but they're oxidized glycerol. Dihydroxyacetone is oxidized glycerol, and so is glyceraldehyde is oxidized glycerol. So animals can produce glucose from sugars or proteins. Sugars include pyruvate, lactate, and oxaloacetate, and protein from amino acids that can be converted to pyruvate or oxaloacetate or some of the other TCA intermediates. They're called glucogenic amino acids. We cannot produce glucose from fatty acids. So production of fatty acid degradation is acetyl-CoA. Cannot have net conversion of acetyl-CoA to oxaloacetate. We don't do that. Plants, yeast, and many bacteria can do that, and they can make anything out of anything, I guess. They can produce glucose from fatty acids, among other things. They can produce everything they need, basically, right? If they have to, right? So glucogenic amino acids grouped by site of entry. So we have pyruvate, alanine, cysteine, glycine, serine, threonine. All of these can be made into sugar. I'm not going to ask you about those just yet. And then we have the pentose phosphate pathway. So pentose phosphate pathway, basically we take, what's the purpose of this? Why do we do this? So
DNA, RNA.
yeah, so we need to have ribose around so we make our nucleic acids. So we start off with glucose 6-phosphate. That's going to be oxidized to 6-phosphogluconate. So gluconate, so it's an acid there. It's gone. And then then that gets decarboxylated to produce ribulose 5-phosphate, and that's where the one sugar goes, right? It's decarboxylated. And then ribose 5-phosphate will then be used to make nucleotides and coenzymes and DNA and RNA, etc. So the main products of the pentose phosphate pathway are NADPH and ribose 5-phosphate. What do you think the difference is between NADH and NADPH?
Phosphate.
Yeah, one phosphate group. But they have big differences in where they're used in the cells. What's NADPH used for? Huh? It's used for anabolism, so it's used for biosynthesis. So you use NADPH when you're making things in the cells, such as biosynthesis of fatty acids and steroids. You'll see that's where the reducing equivalents come from. And then also repair of oxidative damage. Ribose 5-phosphates, biosynthetic precursor of nucleotides, because the 5-carbon sugar, it's the backbone of all our nucleotides. And also the synthesis of some coenzymes. So the oxidative phase generates NADPH from NADP. So we have glucose 6-phosphate. Then gets oxidized to produce this 6-phosphogluconic gamma lactone. Water will come in as lactinase, and you produce 6-phosphogluconate. And so you have this carboxylic acid group. And that carbon will then be the group that leaves after it's decarboxylated. Right? Ow, my nipples. So non-oxidative phase regenerates glucose 6-phosphate from ribose 5-phosphate, used in tissues requiring more NADPH than ribose 5-phosphate. So, some tissues, when you have to store energy, they'll take acetyl-CoA and make fatty acids out of it. And you need a lot of NADPH around for that. So you have ribose 5-phosphate, gets converted to xylose 5-phosphate. Trans-ketolase will convert that to glyceraldehyde 3-phosphate, to trans-aldolase erythrose 4-phosphate, to then to fructose 6-phosphate. And then on the other term, if you isomerize that, you have, don't worry about memorizing these pathways because I'm not going to test you heavily on these at all. So just know the basics for what's important about the pentose phosphate shunt. And that's to make ribose and NADPH, basically. So NADPH regulates partitioning into glycolysis versus the pentose phosphate shunt. So if you have a lot of NADPH around, it's going to block using the glucose 6-phosphate into the pentose phosphate shunt. Because unless you need pentose phosphates, NADPH, high levels of NADPH is going to inhibit producing more NADPH, right? Does anybody know what kind of inhibition this is called?
Substrate inhibition.
So feedback. Feedback inhibition. Desensitization is where you have a receptor that then gets internalized and it gets taken out of that pool. This is actually enzymatic, where one of the end products of a long pathway inhibits the entry into that pathway. So feedback inhibition. So the products feeding back into that first, modulating that first enzyme into the pathway. So glucose 6-phosphate dehydrogenase deficiency causes disease. And I'm not going to ask you about this either. So we can jump to chapter 15. Hopefully we can get it all done next Monday.
Next Monday.
Right? 15 and 16. Huh?
If you don't get through 16, is it hop on to the next?
We'll get through it.
Yeah.
I promise.
It's just citric acid?
Huh?
Citric acid?
Yeah.
Okay.
Yeah.
So. But this is an important one, regulation. We've already touched on some of it, right?
I think a lot of it's just making sure you understand the metabolic pathways and their interactions, what enzymes would react with specific combinations.
Yeah, and we know gluconeogenesis and glycolysis are reversibly regulated, or not, because you don't want that futile cycle. And they're heavily regulated at those steps that have large free energy changes. Everything else is just equilibrium regulated, right? Because they're near equilibrium. You have buildup of products, it'll run in reverse. Buildup of substrates, it'll run forward.
Is there mechanisms that run substrates or that run enzymes between two pathways that are unrelated? Like hydrolysis between the liver and the kidneys.
Like the Cori cycle?
Right.
Yeah.
Not necessarily the Cori cycle. You the liver and the liver and the kidneys run the same pathways. One's the gluconeogenesis, the other one's the glucogenesis. So they have the same enzymes.
Yeah, so like I said, the kidneys is the only other organ that can do gluconeogenesis because it has that glucose-6-phosphatase activity, but the majority of it's in your liver.
Okay.
So, but anyways, so key topics: principles of regulation in biological systems, glycolysis versus gluconeogenesis, and the chemistry and regulation of glycogen metabolism. So biochemical reactions in a living cell or metabolisms organized into metabolic pathways. Pathways have dedicated purposes, which is going to be extraction of energy or storage of energy, right? And that's glycolysis versus gluconeogenesis there. Synthesis of important building blocks, such as the pentose phosphate shunt. We need that NADPH for synthesis of fatty acids and other things, and also the ribose 5-phosphate for DNA and RNA. And then the elimination of waste materials. So the pathways can be represented as a map, and I don't know if you've seen these maps before. They look like this, where they have everything connected. So this is what the extra credit on the test will be if you can map this out. Just with every enzyme possible and all substrates and intermediates. Then you don't have to bother coming to class ever again. No, I'm just joking.
It's on the website. I did look at the website and did a quick bunch of
Yeah, but I hate just memorizing things anyway. So homeostasis, what is that? It's like maintaining balance. So organisms, we're going to maintain balance by keeping concentrations of most metabolites at some steady state. The amount that's used is going to be the amount created. That's going to be that way until, you have some kind of perturbation. So pathways are at steady state unless perturbed. And perturbation will then result in some kind of new steady state, right? So principles of regulation: the flow of metabolites through a pathway is regulated or maintain homeostasis. Sometimes the levels required metabolites must be altered very rapidly, so you need that rapid alteration. Stop burning sugar, and you better start storing sugar because you got plenty of energy around and all this fuel that just needs to be stored. Need to increase capacity of glycolysis during action, so that could happen if, fight or flight syndrome. There's a hormone signal that triggers you to burn sugar and triggers you to release glucose from glycogen stores. Likewise, the reverse is true after successful action. If you have all this high blood sugar now from the action of epinephrine, insulin's going to need to be there and tell you to store all that energy you're not using anymore because you've escaped or beat the thing that was trying to eat you. So, and yes.
How fast does the glycolysis happen in the body?
It's, I don't know the number, but it's pretty fast. Yeah, really fast.
Okay.
But
not like a couple of minutes, but like rapidly, like within seconds maybe?
Yeah, because it's all enzymatically done, so it's pretty fast.
Would you say it's faster than like getting it from like a fat store? Because the fat store, like if they're going to turn it into ketone body too.
Yeah, so that'll have to be converted to ketone bodies to be utilized, because remember, you can't make sugar out of the free fatty acids. You can out of the glycerol.
Okay, so that would probably be a slower process.
Yeah. Yeah, that's why glucose is partially oxidized already, right? Versus free fatty acids, which are all reduced. So, but anyways, this is what I was talking about, feedback inhibition, right? So the product in the pathway will inhibit the entry step into that pathway, right? So what's the product of glycolysis? The main product that you're going for in glycolysis? Huh? No. No, well, that's one of them, but what's the main product? Why are you burning this sugar?
Oh, just branded.
ATP. So ATP will inhibit that phosphofructokinase 1, right?
The one cycle, not the whole.
Well, phosphofructokinase 1 is the entry step. That's the committed step into glycolysis. Not the entry step per se, but the committed step. That's, and I have that right there. Nobody, I ask these questions all the time and it's always right there. So rates, this is what you were asking about. Rates of biochemical reactions, they depend on many factors, Lauren.
Okay.
Concentration of the reactants and products in the tissue, activity of the catalyst, concentration of the enzyme, rate of translation versus the rate of degradation of the enzyme. So enzymes are constantly being turned over just like all proteins, so the amount that's being made and the amount that's degraded. Intrinsic activity of the enzyme, and that could depend on substrates and effectors in phosphorylation. So allosteric regulation is going to affect this rate too, right? Concentration of effectors, so we have allosteric regulators affect that, competing substrates, and then pH and ionic environment is going to affect that, and the temperature, of course. So, and this is that figure showing you the overall things that will alter the rates. So, I don't know, kind of noisy.
Yeah, it does. Once it gets to the enzyme and substrate, it's just cyclic memorization cycle.
Yeah, but then you got to produce the enzyme, so this is where the enzyme is being produced, and that's dependent on how much DNA transcription is making, how many mRNA molecules you have, how fast that's being degraded, how many are being produced, and then the thing about making the RNA is always determined by extracellular signals and transcription factors to turn those genes on, and so forth. And then you have regulatory proteins, which will block the enzyme, kinase reactions and phosphatase reactions, and then I guess this is the active form of the enzyme. And then you have allosteric effectors that'll take free enzyme away. And you know, remember the tight versus relaxed state. So
once you get to the enzymes, the control over what subsequent product is, just what's inside the cell.
yeah, basically what's inside the cell, are you're going to have a regulatory protein that might be controlled by some extracellular signal to phosphorylate the enzyme and turn it off, basically, right? Or there's allosteric effectors that are produced by other enzymes that can take this enzyme out and freeze it in that, or not freeze it, but shift the equilibrium to that tense state. So a lot of things. And then the turnover of the enzyme itself. So rate of the reaction depends on the concentration of substrates. So the rate is more sensitive to concentration at low concentration, right? So if you have a lot of enzyme there, the more, that's a logarithmic scale, right? Remember it looks something like this. So your substrate concentration here, and then boom, and then this is the I don't know, like the appearance of the product concentration. My handwriting's terrible. So you remember, and then, so at low concentrations, a little increase in substrate really has a massive change in the rate at which you're producing the product, right? Maybe I just write rate. I put like the D, that should be dP over dt, right? But yeah, so that's the rate. So at really high levels of substrate, right, when you change substrate concentration, you don't really get much change in the rate. And why is that?
Well, once it reaches equilibrium, there's no more product to be made.
So you have this V max here. It's not that there's no more product to be made. It's when you add more substrate, it doesn't change the rate at which the product is made very much, or at all, really, when you're over here. And that's because your enzymes are saturated. You have excess substrate, then you have enzymes. But when you have low enough substrate concentration, little change in substrate greatly increases your rate. So, and I just had to go to the next slide. I wasted my time drawing. So, but if you look, Km versus metabolite concentrations, and we see here that many of the enzymes have Km that's near or greater than the physiological concentration of their substrate. Why is that? Do you guys remember what Km equals over here? Where do we find the Km? Yeah, 1/2 the V max is the concentration, that's your Km, right? So if most enzymes in your cell, like they say here, have a Km that's near or greater than so most of your substrate concentrations are like around here, right? So they'll be greatly influenced by the concentration of substrate that you have around. Does that make sense? So.
The metabolic concentrations is outlined. That's this, I guess, equation or graph. When I look at it in the book, I couldn't see the relationship between the two. Because I kind of pictured that the Km might be different, even though it's just concentration at a
Kms can change based on concentrations when the enzymes are allosterically regulated, right? But not all this is trying to show you is this, that the majority of enzymes and biochemical pathways, the substrate concentrations are near the Km, which means you're at this part of this curve, right? So you're not saturated, and that just means a little change in substrate concentration will mean a big change in rate of product formation. So this graph it's just showing you that relationship with all these different pathways. They're all pretty much near Km values, substrate constant, okay? So don't try to read too much into that graph. That's all it's trying to tell you. So like we know, phosphorylation of enzymes affects their activity. And so phosphorylation occurs pretty much at serine, threonine, and tyrosine residues because they all have this OH group. And protein kinases will add the phosphate, protein phosphatases remove them. We know that. So that's basically all that slide is telling you. But we already knew that, right? We've been talking about that since the beginning. And enzymes are also regulated by regulatory protein. What was the one that we talked about the other day? Is it protein kinase A, was it? During the cell signaling, it's a quaternary structure in the inactive form where it has 2 regulatory domains and 2 active domains. And when the regulatory domains, I think, do they get phosphorylated, they get removed from the active domain, and then those go in and phosphorylate other proteins and ultimately result in, gene transcription, right? So similar thing here, there's regulatory domains that they're going to regulate the activity. And then proteins have a finite lifespan. So the proteins in your body, they're not going to be around forever. There's a certain amount of turnover. And at homeostasis, the amount degraded is going to be equal to the amount produced, right? Or the rate degraded. So some proteins are as old as you are, crystallines, and like the islands, things like that. And that's why you get old, your vision goes bad. To see you young? I'm still waiting. So reactions are far from equilibrium are common points of regulation, right? So in glycolysis, those ones that have the large delta Gs are always far from equilibrium. So you have that large free energy change. Within metabolic pathways, most reactions operate near equilibrium, right? So all the reactions that are used for gluconeogenesis and glycolysis, those are the ones that are near equilibrium. So the directionality of those is going to be based on the substrate or product concentration, and that's governed by which direction that process is going on in the cell. So, and then these are some tables from your book and showing you basically the equilibrium rates and the mass action ratios and which ones are near equilibrium. So the regulated steps, hexokinase, phosphofructokinase 1, not near equilibrium. Large free energy, right? And then the non-regulated ones, such as aldolase, that splits it into the 2,3 carbon sugars, Triose phosphate isomerase, glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, All of these are near equilibrium, so they flow both ways. Lower free energies, except for that glyceraldehyde, 3-phosphate dehydrogenase, glycerol kinase. Phosphoglycerate mutase, enolase, pyruvate kinase, if you remember, is heavily regulated and it's not near equilibrium. So that's all this is trying to show you. And then for the pyruvate carboxylase and phosphoenolpyruvate carboxykinase again, that's gluconeogenesis, taking pyruvate to make PEP, and that's heavily regulated, not near equilibrium also. So, and then glucose 6-phosphatase, that's near equilibrium, but you're only going to find this in the liver and in the kidneys. And then it's actually in a vesicle, that in the membrane vesicle, that the glucose binds to the enzyme. It removes the phosphate, then it kicks it into a vesicle that'll then, an exosome. It'll turn into an exosome, and that glucose will be packaged in there and fused with the plasma membrane of liver cells and released into the bloodstream from that endosome, exosome rather. So ATP and AMP are the key cellular regulators. Remember, so the ATP is that ultimate product. So a 10% decrease in ATP greatly affects the activity of ATP utilization in the cells, just a small change. And a 10% decrease in ATP leads to a dramatic increase in AMP. So that can be a more dramatic indicator of energy state of the cell. And this little table shows you that. So if you have ATP depletion, 5 millimolar yields 0.1 millimolar AMP. If it's depleted from here to here, you have this great increase, a 6-fold or 600% increase in AMP. So you're going to see a lot of these regulated steps are regulated by AMP concentrations in the cell. So what do you think? AMP, is it going to activate phosphofructokinase 1 or inhibit it? Right, so AMP is a strong allosteric activator of phosphofructokinase 1. And then AMP differentially affects many pathways in different tissues via AMPK. What do you think that is? Yeah. So some enzymes in the pathway limit the flux of other metabolites. Enzymes that are not far from equilibrium are regulated. Not all regulated enzymes have the same effect on the entire pathway. Some control flux through the pathway. Others are going to regulate steady state concentrations of metabolites responses to changes in flux. Hexokinase and phosphofructokinase are appropriate targets for regulation of glycolytic flux. Since the increased hexokinase activity enables activation of glucose, the increased phosphofructokinase activity is going to enables that catabolism of activated glucose via glycolysis. So that's why you get regulated at those steps. And then I think we're about out of time, right? So we made it to 21. So we'll try to finish the next 2 chapters next time and then do the test. Any questions about anything?
Mechanisms do Monday or Wednesday?
I'd give them Wednesday.
Okay, it's a little bit Monday, but I just wanted to.
Yeah, I'm always flexible about due dates, but okay.